for the participants who don't know Deepak or may not know him well, Dr. Deepak Modi is presently working as scientist G and head of molecular and cellular biology lab at National Institute of Research in Reproductive and Child Health, which is an ICMR institute at Mumbai. His research interests
Amrita,
encompass. Now it's mute.
please.
So his research interests encompass understanding mechanisms that drive embryo implantation and placental development, determining causes of implantation failure and pregnancy-related disorders like preterm birth and mother-to-child transmission of infections, development of 3D organ-on-chip and organoid models of reproductive tissues, genetics of sexual development and infertility. He was a part of the team that investigated the effect of COVID-19 on embryo implantation and other aspects of reproductive research. He's a fellow of National Academy of Sciences, India. He received that fellowship in 2020. A recipient of GP Talwar Middle Career Scientist Award in 2017, ICMR Tilak Venkoba Rao Award in 2010, GP Talwar Young Scientist Award in 2009, Innovative Young Biotechnologist Award in 2007, and Young Scientist Platinum Jubilee Award in 2007, to name a few. He has currently 5 ongoing granted projects, which I could retrieve from the his web link, and 6 completed project to his credit. He's an editorial board member of Fertility Science and Research, Human Reproduction, and Journal of Assisted Reproduction and Genetics. And this was just a very short introduction of Dr. Deepak. And I got this opportunity to listen to him on a couple of events. And I was just a few days back telling my students about uh, um, Deepak was in one of the seminars. He was uh, uh, teaching how there is a kind of war between the embryo and the endometrium. So that is a very famous talk. I think Deepak delivered that talk couple of times. The war between the embryo and the endometrium, and I got this opportunity to discuss with my students those findings. So, without delaying, I would just hand over the baton to Deepak.
Thank you so much, Ashwini, for this generous introduction. And thank you, Gopal sir,
Pleasure is all mine, Deepak. Always.
and thank you, Gopal sir, for inviting me. It is an honor to be on this platform. To be
We are grateful. We are
I'm
grateful.
really grateful to you for making me a part of this family. And I'll start my presentation. I think we are recording it, right, sir?
Yes.
Okay. So I will start my slides. I hope I can share them. And I hope you can see my slides and then the full screen mode.
Yes.
Thank you so much.
Yes. Hide. नीचे का line hide कर दीजिए तो. Yes.
Yes. Okay. So the topic I'm going to speak on is about the shrinking Y chromosome. And I come from the National Institute for Research in Reproductive and Child Health. It's one of the ICMR Institute, as the name suggests. We conduct research all related to reproductive health. And in my lab, we are interested in understanding the female reproduction and the male reproduction. I'm a geneticist by training. And we look at fundamental research that goes on in my lab is understanding embryo implantation and early placentation, how things go wrong there in these processes to lead to infertility, recurrent implantation failure, recurrent miscarriages. And if we understand this, we hope we should be able to help the infertile couples. I have a big team of students and postdocs, and I am really grateful to them for doing all of this hard work. And I stand before you just presenting some of the work that they are doing. Our interest is also in the genetics of male and female infertility, and some of this work has gone into translation. And the work that I'm going to talk to you today is done by my 2 graduate students, Shadan and Sanjukta, who have graduated a couple of years ago and was carried forward by my postdoctoral and then the DHR woman scientist in my lab, Dr. Stacey Colasso. And I acknowledge all their hard work. And it is just that they have been the background players. I'm the forerunner today. Let me begin my talk. So we live in a sexually dimorphic world. We identify ourselves as males and females, and both these species, males and females, are phenotypically, genetically, hormonally, and psychologically very divergent. And we see this right from fishes, reptiles, birds, and mammals. But when we begin our early development, we start as a single egg, as a single fertilized egg, which is one cell, which divides and divides and divides and divides to form the blastocyst. And it is this stage at which it implants into the maternal uterus and giving rise to the full-blown embryo. And here is the live development of the human blastocyst. And you can see it's reached up to the blastocyst stage and it has started to hatch. Now, but whatever species that you are on, we all go through these processes of development. However, at this point, we are neither morphologically, sexually dimorphic. However, in our mother's wombs, we were all like this, but when we come out at the end of 9 months, we are get a handsome man or a beautiful woman, or you can even get them. The question is, how are the two sexes determined? What are the genetic and the molecular players that are responsible for developing the males and the females? So we all know it's genetics. Let me take back to your fundamental biology class, which you must have learned in school or first year college, where all the chromosomes, the humans are 46 chromosomes, and when an egg is fertilized with a Y-bearing sperm, you get a 46XY, or if its egg is fertilized with an X-bearing sperm, you get 46XX. If you're fertilized with a Y-bearing sperm, you develop testicles and you become a handsome man. And if you're fertilized with the X-bearing sperm, you're 46XX, you develop ovaries and you become a beautiful woman. This is very simple, fundamental principle of sex determination. And therefore, it was believed that the Y chromosome should carry the genes which are responsible for sex determination. So if we look at the human genome, this is the phenotypic view of the human genome under a microscope. Of course, these are colored with different probes, and you can see all the chromosomes are in pairs, except for the sex chromosomes. In the females, as you have two X chromosomes which are identical pairs. However, the males have a discordant pair where there is one X chromosome and one Y chromosome. And it is on this Y chromosome it was believed that the genes responsible for maleness are there. But however, beyond its role in sex determination, the role of Y chromosome has been largely ignored. So all of us who have been in our mother's wombs develop as bipotential gonad, and these bipotential gonads either form into an ovary or form into the testis. And this testicular determination pathway is because of the presence of the SRY gene on the Y chromosome, which drives the bipotential gonad into the testis. And if the Y chromosome is not activated, the gonad forms into an ovary. So it is a genetic switch, and it was thought that the Y chromosome plays a central role in the sex determination. And beyond this, the Y chromosome does not have any role. And trust me, it is one of the most ignored chromosomes. Half of the people who are listening to this talk have this chromosome, but it is believed to be the most ignored chromosome. And you can see here, these are published genome-wide association studies for different kinds of diseases and to what they are associated. And each of this bar that you can see is one of the human chromosomes. And you can see almost all sorts of genes and diseases are associated with almost all the chromosomes except for the Y chromosome. There is not even one disease reported to be associated with Y chromosome. It's not that there is no association. It is people that believe that the Y chromosome is useless, and it has no roles beyond sex determination, and therefore in these Jiva studies, Y-linked genes are not even studied. And to the extent, the amount of insult that the Y chromosome has experienced is that there have been people who are even talking that the Y chromosome is going to disappear. It is shrinking in the course of evolution. In few years' time, the Y chromosome is completely going to disappear. And if you do a Google search, this is a Google search done by this morning, and where I just said disappearing of the Y chromosome, and in 2024, Business Standard reported about it. The Week had reported about it. India Today, Times of India, Economic Times, almost every leading newspaper had carried this article about the Y chromosome might just disappear. That's going to lead to the extinction of the humans and of the man. And here there is an Instagram post where they said Y chromosomes are declining and will men go extinct. And there are posts which says men are going to be in trouble because they're going to disappear from the planet because the Y chromosome is going to disappear completely. And pi by 2 Y chromosome. So there's so much of interest, and this is largely because there are a group of scientists who believe that the Y chromosome is actually going to disappear. And the idea of this entire disappearance of the Y chromosome have come from studies from Professor Jennifer Marshall Graves in the Australian National University, who has been studying the Y chromosome from different species, especially from the platypus. And she believes that the Y chromosome is shrinking in the course of evolution, and at some point it's going to disappear. And by her calculations and all the others who have proposed it, it is believed that the Y chromosome is going to go extinct in nearly 4.6 million years from now. Now the question is, why do we think that the Y chromosome will disappear? There's several reasons for us to believe in that. But before that, we need to understand how the Y chromosome has evolved. So several million years ago in the timeline, I can tell you about 350 million years ago, the sex chromosomes were identical pairs, whether you were male or a female. There were no specific sex chromosomes. However, due to a random mutation on one of the chromosomes, there was the evolution of the SRY gene, which happened somewhere around 240 to 320 million years ago, where where the first mutation happened, which led to the development of SRY gene. And around the SRY gene, more genes which had sex-specific functions started accumulating, giving the evolution of the Y chromosome. Then this Y chromosome underwent several inversion events. So there were about 4 inversion events which occurred during the period of between 320 years to about 50 million years ago. And the present generation Y has evolved, which has no homologue to the X. So these originally, if you can imagine, are the X chromosomes. The SRY evolved, but the rest of the chromosomes were homologous to each other. But over a period of time, because of the inversion events, deletions, and duplications, and adding new genes from the other autosomes, the Y chromosome evolved, and the present day Y chromosome has very little homology to its other sex chromosome, except for the pseudo-autosomal regions. So essentially, the Y chromosome has evolved out of the autosomes, but has now, in the current period of time, acquired a very unique set of sequences in the humans. So, and if we look at the sequence similarity across primates, this is from bonobo to orangutan to humans, you can see that the X chromosome is very closely homologous in all species. So between, let us say, human to bonobo, it is 94% identical, and human to gorilla, it is 85% identical. So within the primates, there is strong sequence similarity on the X chromosome, but there is a very strong sequence divergence on the Y chromosome. The human Y chromosome is only half as similar to its closest relative chimpanzee, and its gorilla is just 25% similar. So the similarity of the human Y chromosome across species is less than 30%, except for the chimpanzee, which is its closest homologue, which is its half. So the Y chromosome, it appears that the Y chromosome is evolved at a very rapid rate, and it is very divergent. So it is believed that when a chromosome which is evolving at a very rapid rate, it tends to acquire mutations. Number one. Number two, the autosomes correct the mutations because during the meiotic crossover, they have a sister chromosome which acts like the original template, and it looks at the original template and corrects itself on the other chromosome. So the correction mechanisms happen. Now, as I told you in my previous slide, that the human X and the Y chromosome are so divergent that they just do not pair in meiosis. They cannot pair in meiosis, and since meiotic pairing cannot happen, it is not going to have a repair mechanism, and deleterious mutations which have acquired on the Y chromosome will keep on accumulating, and at some point, because of these deletions, because of these deleterious mutations being accumulated, and more damaged genes keep coming on the Y chromosome, the genes keep on getting lost, and the rate at which the genes have got lost, if I based on the calculations which have been proposed by Mueller's, it is called as a Mueller's ratchet. The chromosome will disappear in about 4.6 million years from today. And if we look at this, there are very few shared genes, as I told you, between the human and the chimpanzee. So the reasons that have been proposed for the disappearance of the Y chromosome are because most genes on the modern Y chromosome have completely disappeared. All those which were there in the original Y chromosome are not there. Some of the genes which are there are completely inactivated or have become pseudogenes. Many of them have acquired mutations, making them less active, and very, very few genes left which are intact and functional. And the functional genes are largely proposed to have a role in sex determination. And let me remind you that sex determination is not just dependent on the Y chromosome. There are many other genes on the autosomes which do it in many species. And therefore, since a function of sex determination, that is forming a male and a female, can be done by other genes on the autosomes, it is believed that if the Y chromosome disappeared, nothing happens. And it is believed that the role of other genes on the Y chromosome is very restrictive and redundant. And the proof to this has come from two living and fertile mammals without the Y chromosome: the spiny rat in Japan and the mole vole are the two species which do not are two mammalian species which are fertile, happily reproducing in the capture as well as in the wild, do not have a Y chromosome, and the autosomes do the job. So this has been the premise to which has been believed that if the Y chromosome will disappear from the human genome at some point, and we will start doing sex determination by alternate mechanisms. However, the question that I asked is that why the Y chromosome? Will the Y chromosome really disappear? Or is there an alternate hypothesis to this possibility? So I do not think that the Y chromosome will disappear because of four reasons, and I'll give you evidences of my theory that I have for disappearance, for not the Y chromosome to not to disappear. Primarily, up to now, our understanding of the human Y chromosome has been incomplete. The Y chromosome genes are not just responsible for sex determination, but these genes are critical for spermatogenesis. Deletion of these genes, if you start losing these genes, it causes infertility in humans, so the reproduction of the species will be compromised. And the Y chromosome genes do not just have a role in infertility, but they have a role in tissues beyond the gonads, and they have roles beyond fertility. And I'll present evidences for all of these in my next coming slides. So as I told you that the Y chromosome has a role in sex determination, and the job of this is on the SRY gene, which is on the short arm of the Y chromosome. And for years and years, it was believed that the long arm of the Y chromosome, which does not recombine with the X, is the useless portion of Y, in it as junk DNA, which does not encode for any genes. However, it was exactly 20 years from today, it was 20 years or maybe a little longer, 1997, in Science, two authors, Bruce Lehane and David Page, published a paper where they claimed they have identified 12 novel genes or families with 10 full-length complementary DNA sequences in the non-recombining region of Y. When I use the term non-recombining region, it is also called as the male-specific Y or MSY. So where only the SRY gene was identified, about 20 years ago, 12 additional genes were identified on this chromosome, giving the first evidence that the Y chromosome is not just a junk box or a carrier for the SRY. It does have a lot of genes and gene families which are expressed in the testis. Very recently, in August 2023, the complete sequence of the human Y chromosome was published, and this paper tells us that beyond what we knew after the sequencing of the human Y chromosome, 41 additional protein coding genes were identified, which we didn't even know existed in our genomes, because the sequence of the Y chromosome was not complete to date, and it's only after the T to T of the human Y chromosome that we have discovered. And what is our 2023 understanding of the Y chromosome? Don't bother about this table too much, but in table 1 of this paper, what they've suggested is that there are 693 genes. Of course, as compared to the X chromosome, which is way bigger, has more than 1,000 genes, there are 693 genes on the Y chromosome. About 106 are protein coding. There are 110 additional genes which have been identified, of which 41 are protein coding. They make more than 1,000 plus transcripts because of alternate splicing. And as compared to the earlier Y chromosome which has been sequenced, a large numbers of new genes have been identified. And this is the ampliconic gene copy which I'm going to talk to you about a little later. About 164 additional copies of the TSPY gene and 3 additional copies of RBM gene, Y gene was discovered. So the proponents of the Y chromosome disappearing theory were those who believe that there are no genes on the Y chromosome. Actually, now there is evidence gathering to show that there are genes on the Y chromosome. So in my lab, we have been interested in understanding what are the roles of these Y chromosome genes. Of course, SRY, a lot of people, including my own work during PhD, we had shown the roles of SRY in sex determination and a number of others. There are others who are looking at the TSPY gene family. We focused on a more smaller region of the Y chromosome, the YQ, which is shown in light peach here, which is divided into 3 locus: AZFA, AZFB, and AZFC. And in total of this AZF locus, I'll tell you a little later why we were interested in this locus. In this AZF A, B, and C locus, there are a total of 244 genes, 68 of which are protein coding. 20 of these are transcribed but not translated in the testis, meaning that they're non-coding RNA and they would have other functions, and about 188 are pseudogenes. So we focus on the long arm of the Y chromosome, and we first try to determine, are the genes on the long arm of the Y chromosome within the AZFC locus active? Indeed, this is one of the gene called as RBMY, which is present on the Y chromosome AZFB locus. This is in human spermatogenesis. You can see these are spermatogonia, spermatocytes, packetine, round spermatids, elongating spermatids, and elongating sperm. The RBMY gene, which is labeled in green here, is nicely expressed in the spermatogonia. It is there in the spermatocytes. It is there in packetine cells. It is there in round and elongating sperm. We could also, this is in the section of the testis. We also looked at another gene, DAS, which is nicely expressed in the germ cells of the human testis. And of the genes which are known on the AZF locus, we found that almost all of them, except for one which is PRY, almost all of them are abundantly expressed in the testis. So we believe that the 98% of genes which are there on the AZF A, B, and C locus, which is a very small region on the Y chromosome, are transcribed. If they are transcribed and the proteins are made, means they should have functions. So what are the biological functions of these AZF genes on the Y chromosome? So if we start using softwares and predict their functions, most of these have a predicted function in spermatogenesis, which is expected. But in addition, they have functions in regulation of gene expression. They have functions in regulation of chromatin organization based on the similarity to other genes in these pathways. They have a regulation in gene translation and protein synthesis, and they also have regulation in mRNA processing. And similarly, we could predict the molecular functions of these genes, implying that the Y chromosome genes not only have just functions in spermatogenesis, but have more ubiquitous and housekeeping functions like regulation of gene expression, protein translation, and chromatin organization. That means these genes are not just there, they are expressed during spermatogenesis, and they don't just have a function in spermatogenesis, but they're very specific molecular functions. So the Y chromosome genes are not just, we are now understanding our whole Y chromosome, but these genes are critical for spermatogenesis. And if these genes are deleted, I will now show you data that the deletion of these genes cause infertility. So I told you we were interested in this AZF A, B, and C locus because about 25 years ago, people started reporting that males who are infertile have either the AZF A, AZF B, or AZF C locus deleted. So they were doing deletion mapping of the human Y chromosome, and they looked at a bunch of infertile patients, and they found that men who do not have spermatogenesis, these regions get deleted, and they are referred to as Y chromosome microdeletions. So in my lab, we started asking what are the contributions of the Y chromosome microdeletions in the Indian population. So Shadan and Sanjukta started this work in my lab with the help of Dr. Jyotsna Gokral, who was a collaborator at that time. We collected data of 403 men who either did not make sperm, who did not have sperm in the seminal plasma, they're called as azoospermic, or who had low sperm count, or low sperm count and poor motility. And when we look at the presence of these Y chromosome microdeletions, we found that in out of 403 men, in about 4.9% of them, there were Y chromosome microdeletions. About 6.4% of men with azoospermia had Y chromosome microdeletions. Almost a similar number was for the oligozoospermia, and about 3% of men with oligosteroteratozoospermia, there was loss of Y chromosome, portions of Y chromosome, indicating that if these genes are deleted, or these large chunks of the DNA are deleted, it will predispose this men to infertility. We never found these Y chromosome microdeletions in normozoospermic men. So the men who have normal spermatogenesis always had an intact Y chromosome, but men who had deleted spermatogenesis, about 5% of them had Y chromosome microdeletions. So then we asked what happened to the 95% of them? Why were they not making sperm or making low quality sperm? So we focused on a much shorter region, even still smaller, which is called as a AZFC region. This AZFC region is interesting because it is a palindromic region. That means this region has multiple repeats. You can see that there are genes organized in head-to-head or tail-to-tail or head-to-tail orientation, and there are repeats. So for example, G1 has two local G1, G2, and G3. There are two palindromes of R1, R2, R3, R4, and they are yellow palindromes and they are blue palindromes. These are just colored for your understanding that these are sequences which are repeated across, and this is called as the AZFC locus. Now this AZFC locus, because I told you that the Y chromosome cannot pair and cannot find its partners. So during meiosis, Y chromosome undergoes self-pairing to repair itself. That if there is anything going wrong in one of the gene here, it will find its similar partner on the other and repair itself. Now during this repair, sometimes mistakes happen while doing the correction, and this can lead to inversions or deletions or both. And these are called as GRGR or AZFC partial deletions. So here is the AZFC map which I just showed you. The deletion of the entire locus is called as a AZFC deletion, but there could be a smaller deletion involving more the blue region or the green and the red region, and so on and so forth. These are very complicated deletions. I'm not going to bore you with why these deletions are happening, but we were interested in figuring out if there are deletions, does it predispose these men to infertility? And lo and behold, we collected data from literature, including a lot of data from India and our own, and we classified these patients by fertile and infertile who had oligozoospermia or azoospermia. And we simply looked at prevalence of these GRGR and the B2, B3 deletions. And we found that in the African population, the GRGR deletions are commonly found in the fertile men and at the same frequency in infertile men. But in the Americas and the Asias and the Australia, the GRGR deletions are strongly associated with male infertility, but the B1, B2 deletions are not. In Africa, it is the other way around. The B1, B2, the B2, B3 deletions are associated with infertility, but not in case of the other population. So that means depending on the population now, if you delete a smaller portion of the Y chromosome, there could be infertility. What did we find in our population? We found that about 2% of men who had normal spermatogenesis had the GRGR deletion, but almost 10% of men who had azoospermia or oligozoospermia had the GRGR deletion, but not the B1, B3 or the B2, B3 deletion. Means in the Indian context, the GRGR deletions are as a risk factor for male infertility. And even amongst men who, normospermic men who had the GRGR deletion, there was a significant lower sperm concentration and reduced motility. They did not qualify to be called as infertile or oligospermic as per the WHO classification. But if they have this genetic deletion as compared to the non-deleted counterparts, there is a significantly lower sperm concentration and motility, telling us that indeed these are pathogenic for these regions are necessary for fertility, and if these regions are deleted, it would lead to infertility. Now, I told you that this GRGR locus has several multi-copy genes. So, for example, DAS is present in 4 copies, TTY4 is present in about 4 copies, BPY2 is present in 2 copies, CDY is present in 2 copies. So now we found that about 10% of men have these deletions, which are much larger. Do these copy numbers, so can there be individuals who just have the DAS copy deleted and they are infertile, or who have the CTY1 or the TTY4 copies deleted and they are infertile? So Sanjukta started looking at copy numbers of DAS, CTY1, Golga, BPY2 and TTY4 genes in fertile and infertile men. What did we get? So first of all, she confirmed that even if there is no deletion in men who have azoospermic and do not make sperm, there is lower levels of RBMY and des mRNA. Shadan had done this work in a PhD, and she showed that even if the copy is intact, functionally compromised AZF genes can lead to infertility. Extending this work, what Sanjukta said was that these men have the copies of the DAS or the CDY1 or the BPY2 deleted. And what she found was that not all copy deletions were significantly different between fertile and infertile men. Like, for example, loss of DAS was associated with azoospermia, loss of CDY1 was almost always associated with oligospermia, but not Golga 2P2Y, but Golga 2P3Y deletions were associated with azoospermia, telling us that specific genes, if they are lost, copies which are lost, the other copies do not compensate for it, and it can lead to azoospermia. And here is a map. This is slightly complicated, but I will want to I want to grab your attention here, is that this is the AZFC locus which we were looking at. And this is the orientation of the genes which are present on the Y chromosome from 5' to 3'. The each row is the gene, and each column, the multiple columns here, I'm leaving it to your imaginations, but these are the region. These are all normospermic men, these are all oligospermic men, and these are all azoospermic men. What did we find? We found that there were several genes which were deleted in both normospermic and oligospermic men, and the frequency was even same. For example, CDY1 here you see it is very commonly deleted in normospermic and oligospermic men. DAS 1 and 2 was also commonly deleted. TTY4 was also commonly deleted. But if you look at this block here, rarely we could find normal men who had these deletions, but these deletions were in this block. So these copy deletions were almost more commonly found in men who had oligospermia or azoospermia. Another gene was this Golga 2P3Y. So we call this as minimal critical locus essential for spermatogenesis in AZFC, telling us that if you lose these genes from your Y chromosome, it's not going to compromise your infertility. It is indispensable for fertility. You can live happily without these genes, no problems. But if you start losing genes from this locus, infertility sets in, reproduction will not be possible, and this Y chromosome will go extinct from the human race. So that means that after a point in the course of evolution, when this Y chromosome is evolving, it is deleting the genes. The genes which are deleted and does not compromise fertility will go on in the next generation, will go on in the next generation, so on and so forth. But if these genes which are essential for spermatogenesis get deleted, it will manifest as infertility. There will be no future generation with such a Y chromosome, and these deleterious mutations will be wiped out from the population, and only the cleaner Y chromosomes will be retained. So I think that the Y chromosome is not going to disappear. we don't think that the theory is going to be like this. It looks like that the Y chromosome is possibly pruning. We see evolution happening in front of our eyes, where we see men in whom there is deletion of Y chromosome, but they are living happily here. And there are men who have deletion of the Y chromosome before become infertile. So we think that the Y chromosome currently is also still big enough, harboring a lot of unwanted genes. But after some point, after some point of a rapid decay, it's going to slowly keep decaying till it is going to reach a point where it is going to reach immortal. Positive selection will kick in from here, and only those Y chromosomes which are healthy and keeping the fertility intact will propagate into the race, and the deleted and the deleterious Y chromosomes will not propagate. It will lead to immortal Y, and then after that point, Y chromosome will cease to get shrink and it will continue as an immortal Y. So this is the alternate theory which I propose based on our data. And this is more recently supported by the data from Marco et al. And the paper was published last year in Nature. It's fairly complicated. I don't want you to go through these bars, which show that how from bobonos to humans and orangutans, a Y chromosome has exchanged its genes in the structural map of the Y chromosome. But I want you to pay attention to this graphs. The graphs here show the number of ampliconic genes, the number of ancestral genes, and number of palindromes. The ancestral genes are the genes which are there on the human Y chromosome, since its evolution in the beginning. And there are about several, I think that gene density, this is gene density per megabase, and in the chimpanzee, in the bobono chimpanzee, in the human Y chromosome, there are about 14 ancestral genes, and these ancestral genes are present in bobonos, to chimpanzees, to gorillas, to orangutans, and so on and so forth. So the ancestral genes which are there have not disappeared over a period of time, and these ancestral genes, some of these even belong to this ampliconic region. So if you look at the ampliconic region also, the ampliconic region which I showed you right now has remained fairly constant across evolution. So it is the non-recombining region. Within the non-recombining region, the region which is not required for the survival of the species in terms of its sexual reproduction have disappeared. The regions which are essential have fairly remained constant across species, telling us that the Y chromosome genes are not just critical for spermatogenesis, but if you delete these genes, it is going to cause infertility and therefore extinction of those Y chromosomes. And now I will give you some data to show that the Y chromosome genes have roles in tissues beyond the gonads. But before I go to this part, I'm going to switch gears here and tell you that how this very fundamental research kind of information, which is on the Y chromosome microdeletions and male infertility, has actually helped us to develop a test for the Indian infertile males. What we had found was that how do we know? So when we were doing these diagnostics, for detecting the presence of Y chromosome microdeletions in infertile males, we had to do PCR. And this PCR required STS markers. Now, different studies across the world, including several studies in India done by our lab and other collaborators, had found that this Y chromosome, the testing for this Y chromosome, they'd used different sets of STS markers, and each of these groups had used different sets of STS markers. So, and we found that if you use fewer STS markers, you miss out on a lot of patients. If you use more STS markers, you are extra looking at the Y chromosome, and you really don't need so many markers. So what Stacey had done as a part of her postdoctoral research in my lab, she carried out meta-analysis of more than 90 markers which have been evaluated in the Indian population. She applied statistics and identified 16 markers. I'm sorry, it's written 14 here, but there are 16 markers which she identified, which she thought were diagnostic for the Indian population. She developed a multiplex-based PCR test, which underwent several rounds of testing within the lab. And finally, we had this multiplex PCR assay, where you can test the presence of these deletions by doing a PCR. So you isolate DNA from infertile infertile men, you do a PCR for the markers for AZFA, AZFB, AZFC, GR, because I told you GR is crucial for male infertility in the Indian context at least. And these are the markers which we find are most commonly deleted in Indian infertile men. And you simply count the numbers of bands in each lane, and if the bands absent or present, you can diagnose the genetic cause of male infertility. And this is an example here. The patient PB here has all the necessary bands for the AZFA, AZFB, and AZFC, but instead of two bands for GRGR, there is only one band, indicating that there is a very small deletion. The patient is labeled having GRGR deletion, and is the infertility is because of this GRGR deletion. Similarly, patient GS has no bands in the AZFB lane, no bands in the AZFC lane, and there is a missing band in the GRGR lane, telling us that the patient is a complex AZFB, AZFC, and a GRGR subdeletion, indicating that this is a test which is very rapid. We have tested this kit assay with a number of labs, and we currently found that earlier whatever data was, we had used 6 markers which was proposed by the European population to detect the presence of Y chromosome. We had got the frequency of 5%. And now when we evaluate these patients, we find that the frequency is 10%, because in the earlier studies, we had missed out a lot of patients. In the European population, 6 markers were found to be sufficient. But in the Indian population, we find the 6 markers are not sufficient, and we need additional markers. So we have developed this test with additional markers, and I'm happy to tell you that this technology, which was developed by us through ICMR, has been transferred to a lab who is now selling this assay commercially, so that those who are interested can buy this assay. And diagnostic labs currently are utilizing this assay and giving now more accurate diagnosis for the patients, unlike the European markers, which were suboptimal for our population. And we have taken this technology from technology readiness level 3, where Stacey had started in 2019. We took it to technology readiness level 8. And in 2022, we have transferred this technology. It is highly reproducible. We have tested it across labs. It has very low inter and intra-assay variation, and our failure rates are also very low. So we are very excited that how a very fundamental research question which we had started has actually led to a clinically relevant problem, a clinically relevant solution. Currently, this assay is being tested by a lot of my collaborators across India. It's been tried in Jammu, it's tried in Ahmedabad, it's tried in Wardha, it's tried in Bhubaneswar. A number of centers are evaluating it. And we are currently compiling this data to get the final numbers, that is what is the frequency of Y-chromosome microdeletions in the Indian infertile men, and what are the common patterns of these deletions. However, clinically, why testing of this Y-chromosome microdeletion is crucial. Now, when there is a man with infertility who has low sperm count, typically clinicians give a lot of spermatogenesis enhancing drugs like anti-estrogens or testosterones or aromatase inhibitors, gonadotropins. Sometimes they give vitamins, multivitamins, antioxidants to just improve the spermatogenesis. However, those men who have these AZF deletions will not benefit from these therapies. So we suggest that if you do a Y chromosome microdeletion testing, and if the patient has a Y chromosome microdeletion, this patient should not be subjected to any forms of treatment, rather should be directly told that you can undergo IVF using donor sperm, or you can do something called as a testicular sperm extraction. Men who have Y chromosome microdeletions do have residual spermatogenesis. A few of the seminiferous tubules do normal spermatogenesis, but they don't do enough so that enough sperm is fed. But there is residual spermatogenesis. You can extract these sperm by a process called as testicular sperm extraction. Take these sperm and inject into the egg by a process called as ICSI, and both of these are routinely done in IVF labs. You can make embryos. These embryos are now transferred to the mother, and you can make babies. However, these patients who have Y chromosome microdeletions need to be told that all the male babies which will be born to you will also be infertile. And you can see in this one of the pedigrees that the father was infertile who underwent a ICSI and had two sons, both were infertile, oligospermic. And again, this patient 3 went under the also chose to undergo ICSI, had a third generation who was also infertile. So we perpetuate infertility in the generation by doing this process. However, because the females are not affected, many patients feel that, okay, if I have a female child, she will be normal completely because she doesn't have Y chromosome. And many men feel that, okay, I am infertile, you're giving me option of baby, you will also give the option to my male sons. But it is fine, but it is essential that the patients are counseled. So men with AZF deletions will not benefit from hormonal therapies to improve sperm count and motility. Antioxidant therapies will not improve spermatogenesis. So therefore, it is essential that this test be done. And as Y chromosome is inherited from father, all the sons will have 100% transmission of father to son transmission of the deletion, and the sons will be infertile. And this is a key point essential for counseling before offering IVF to these patients. And we have released a policy brief which is present on our website, and if anybody is interested can look at it, of the policy statement on the need for Y chromosome microdeletion testing in male infertility. However, we also found that if the patients undergo a ICSI, this Y chromosome microdeletion profile can help the clinicians to predict the outcomes of IVF. Is the success rate the same? Do these men who have Y chromosome microdeletion, if the sperm is taken from them, do they have a good success rate? Classical textbook knowledge told us that men who have AZF-A or AZF-B deletions will have complete arrest in spermatogenesis and you can never get sperm from them. Only with men with AZF-C deletions have residual spermatogenesis and you can do a IVF in them. However, we have data now to show that this data is wrong, and enough data has been collected from these patients, and now we have shown that all men with AZF-A, AZF-B, and AZF-C deletions have a good frequency of removing sperm from the testis, and they can make the embryos. However, we also found that these genes, remember these are all the genes of the AZF A, B, and C locus, and almost all of these genes are also expressed in early embryos. That means that when these genes are expressed in early embryos, men who will have deletions, their resulting XY embryos will not make these genes and will have compromised quality. Indeed, that's what we found. We found that the sperm retrieval rates with men with AZF-C deletions, for example, are higher. Their fertilization rate, clinical pregnancy rate, and live birth rate are significantly lower. They have a very low cleavage rate. Only half of the embryos will correctly cleave. Only 30% of the total embryos will result in a clinical pregnancy, and only half the embryos will result in a live birth, telling us that the loss of Y chromosome not only causes infertility, but also compromises early embryonic development and resulting in not just infertility because of a male factor, but it will also lead to failure of embryo implantation and not a successful IVF. So then this was all statistics which had applied, and clinically this is challenging because each deletion and each type of copy number variation, we asked that can we predict these outcomes for all types of AZF deletions?
Avijit Sharma, mute your mic. Avijit Sharma. Yes, please continue, sir.
So can we predict the outcomes of these types of deletions using something better? So what we did here was we used artificial intelligence. What Stacy did was she collected data. I'm not sure if I have it. Yes, I don't have the data here to show exactly how we had gone ahead doing this, but we collected data of more than 3,000 men who had Y chromosome microdeletions. We had the clinical data, hormonal data, and IVF data. Professor Abhishek Sen Gupta from Amity University and Priyanka Narad, who was there earlier and now she's at ICMR. Together, we have developed this AI-based, artificial intelligence-based software where we take in the data of the Y chromosome microdeletion and the metadata of the IVF, and we have developed this tool which we call it as Fertility Y Predictor. This is a web-based tool which can predict the Y-chromosome microdeletions based on three different parameters. That is a locus which is deleted. So this is a page where you have it. You enter your patient ID, you enter the patient name, you enter the karyotype and say there's a patient has a AZFC deletion based on the internal control markers. Because it is done using PCR, whichever test you have, you can use it, and you can upload the patient for the gel and click on predict. As soon as you click on predict, it is going to tell, okay, the patient has AZFC deletion. Now, this is no rocket science, but when you click on this, it further gives, based on the patient's metadata that is fed in the software, it tells that this patient has an 84% chance of a sperm retrieval, but has a much lower chance of a successful live birth, has a much lower chance of clinical pregnancy and a successful live birth. I'll show you another example. You can enter the STS markers. So different labs use different STS markers. So you can enter the STS markers which are deleted. You can again upload the gel which is there. And when you click on predict, it says that, okay, they're based on the STS markers which you have entered. The patient has a AZFA deletion, and these are the chances of its successful clinical pregnancy. And similarly, you can do it with the DYS markers as well. So I'm going to skip this in the interest of time. But essentially, this fertility predictor is a tool which allows a prediction of IVF outcomes in patients with microdeletions. This was copyrighted by Amity University, and again, it is licensed to the same company, which is now selling our kits. And those who buy the kit get this software along with them. Clinicians now are using this software to predict the IVF outcomes, the fertilization rate, the clinical pregnancy rate, and live birth rate based on the type of deletion that is entered there. And this helps the clinicians. This paper was published. In fact, this is submitted. I'm happy to let you know that this paper was accepted in December 2024. And here the clinicians are now able to use this tool to predict the outcomes of IVF, and it's helping them for an effective clinical decision making. The patients now don't just rely on arbitrary numbers, but in precisely the numbers are told what is their chance, and they can say that, oh, I have a very low chance of a live birth rate. I don't want to undergo IVF. I'll be happy to go under a donor sperm or opt for an adoption. Or some patients say, no, I have a low chance, but I do have a chance. I want to undergo. So then the clinicians will tell them that you will have to do a lot of cycles. Multiple cycles will be required. So depending on how many cycles it is required and so on and so forth, it helps the clinicians to take this effective decision making. So the take-home message from the work is that the Y chromosome has large numbers of genes such as housekeeping functions. This Y-chromosome microdeletions causes infertility in 10% of cases. The test use must be defined and validated for the Indian population. I told you that the earlier tests which are available are all for the European population. We have developed this test which is valid for Indian population. And once a man with a Y-chromosome microdeletion is identified, he's not going to benefit from hormonal therapies, can be directly asked to undergo IVF ICSI, but he should be counseled that there is going to be a vertical transmission of the genetic defect to all his sons. He will have poor fertilization rates, low good numbers of good quality embryos, and low chance of clinical pregnancy. So this test helps in appropriate clinical decision making, and therefore we believe that it is mandatory to test the Y chromosome microdeletions in Indian infertile males. Having said this, I'm going to move to the final point, which is that why I think that the Y chromosome is not going to disappear, because now I'll show you data that Y chromosome not only has a role in reproduction, but there is emerging evidence now to show that the Y chromosome also has roles in tissues beyond the gonads and may have a role beyond fertility. So what Stacy had done was she looked at the expression map of these genes across all human tissues. About 30, 15 or 18 of them are listed here, ranging from adipose to adrenals to bone marrow to stomach. And you see that the entire Y chromosome AZF locus is expressed in the testis, which is there, but you can see genes in the AZFA and the AZFB locus are also expressed in the appendix, the bone marrow, cerebral cortex, every freaking tissue you see. They are all expressed in the tissues, in the somatic tissues, non-reproductive tissues also express the human Y chromosome, except perhaps the smooth muscles where you don't see any of these genes being expressed. Epididymis, you see one of the AZFB gene expressed, but you can see even in the parathyroid, the pancreas, and even the placenta, the Y chromosome genes are expressed, telling us that the role of Y chromosome is not extus exclusive. And if I need to give you quantitative data, about 30% of Y chromosome genes expressed in the kidney, 34% in the stomach, about 20% in the lung and the heart, 25% in the bladder. So almost many tissues express the human Y chromosome, and it is found that in multiple cancers, for example, in hepatocellular carcinoma, the genes in the AZF locus are either upregulated or downregulated, and the Y chromosome genes are found to, and the loss and the mutations or dysregulation in the expression of Y chromosome genes is reported in lung cancer, prostate cancer, head and neck cancer, renal cancers, and colorectal cancers. So that means that in these cancers, there is dysregulation of the AZFC locus, predisposing them to multiple cancers. More recently, there is a concept coming as mosaic loss of Y. As men start aging, because this Y chromosome is very small, and during the course of cell division, there is something called as mosaic loss of Y, which means that some of the XY pairing cells just abruptly or randomly start losing the Y chromosome and they are XO. So the patient, if you do a FISH or a quantitative Doppler digital PCR, you find that there is reduction in number of cells having the Y chromosome, and this is referred to as the mosaic loss of Y. And this mosaic loss of Y currently has shown to be strongly associated with cardiovascular disease, neurodegenerative disorders like Alzheimer's disease, and cancers as well. And I'm talking about non-reproductive tissue cancers like leukemia and renal cancer. And this has also been shown experimentally in the mouse. They created mice which have loss of Y, mosaic loss of Y. So they took bone marrow cells from the mice, they deleted the Y chromosome using CRISPR-Cas9, and they reconstituted the bone marrow with, and they injected this loss of Y cells into the circulation of these mice, and they followed them over a couple of weeks, and they developed that they found that these LOY cells are now well accepted into the mouse, and they found that as these animals age, there is a high incidence of heart failure. And they also found that there is accelerated, and these mice also had accelerated tumor growth. There was an increase in leukemia, increase in the incidence of leukemia in these animals, and there was enhanced response to anti-PD-PDL1 antibody. And this is a very beautiful paper which was published, I think, earlier last year, where they showed that these mosaic loss of Y, these Y chromosome genes, if they're lost in the monocytes, they land up into the heart, and into the heart they undergo a transformation leading to fibrosis, fibrotic myocardium, eventually leading to cardiac failure. And they found that this is because they create a lot of inflammation locally. The system does not recognize these cells as self, and they start creating inflammation, telling us that the loss of Y chromosome is not just leading to male infertility, but this loss of Y chromosome can also lead to predispose these men to more generic conditions. And I also showed you that the Y chromosome genes, the AZF genes, are expressed in the brain. About 21% of these genes are expressed in the brain. And it is reported, earlier it was reported that we have also found this, that men who have Y chromosome microdeletions as they age develop there is an increased incidence of neuropsychological disorders like bipolar disorder, major depression, and language impairment. And we had this very interesting case report. A father had Y chromosome microdeletion, AZFC deletion, partial AZFC deletion, had severe oligozoospermia, underwent ICSI, and then delivered a male child. His deletion expanded during this process of his spermatogenesis, and this child had mental retardation and developmental delay. delay. So it is important that these men who have Y chromosome microdeletion should be counseled for their health issues, but they also have to be counseled that their children also might have more deleterious health issues. So to summarize, the Y chromosome has multiple roles beyond fertility. I've shown you data that it has role in early post-zygotic divisions. It alters early fertilization events in embryonic development. Of course, Y chromosome genes have a role in sex disorders and sex development. In the young adults in the young adults, Y chromosome has a role in spermatogenesis, and if there's a loss of Y chromosome, there is spermatogenic failure. And with aging, when there is loss of Y chromosome, there is increased predispositions to cancer, Alzheimer's disease, and cardiac disease. So to summarize, I believe that the Y chromosome will not disappear from the human race, but at some point a positive selection will kick in, because when you start losing this Y chromosome, there'll be health issues, and such Y chromosome will be rapidly excluded, because there will not be reproductively viable species, and the critical genes which are there on the Y chromosome will be retained, and the Y will be forever, because these Y genes have critical role in spermatogenesis. They have more ubiquitous functions than previously thought. It has a remarkable ability to repair itself, and deletions of these genes will not only affect fertility, but it has loss of these genes have detrimental effects on men's health, and therefore it is important to retain Y. So all the males who are listening to me, go be happy, go home and have dinner well, because you are not going to disappear from the planet. I thank my collaborators, Dr. Indira Induja and late Dr. Kusum Zaveri, who was strongly instrumental in giving us these samples. I also thank Dr. Reema Dada from AIMS, who has provided us with a lot of data on the Y chromosome. Ashok Pal and Prafulla from MGM Sevagram have also been our long-time collaborators. Dr. Jyotsna Gokraleta Institute originally started this project with me, and I've carried on for the last 15 years. And I also thank Dr. Abhishek Sen Gupta and Priyanka Narad, Amity University, for helping us to develop the AI tool and the network of labs who are validating our assay. Thank you, and I'll be happy to take questions.